Welcome to this short video on the purine synthesis pathway. Our genomic sequences are determined by bases present inside our DNA. These bases come in two types, purines or pyrimidines, and there are two of each. The purines are adenine and guanine, and these pair to thymine and cytosine, respectively. This video will detail the synthesis of these purines. The purine pathway starts with ribose 5-phosphate, synthesized by the pentose phosphate pathway. The first step is 5-phosphoribosyl 1-pyrophosphate synthesis, which we will call PRPP synthesis. As it is inhibited by the purine nucleotide end products, it is a limiting step in purine synthesis. Activated by inorganic phosphate, ATP is used by PRPP synthetase, catalyzing the reaction to produce PRPP. 5-phosphoribosylamine is then synthesized via PRPP, glutamine, and water, being catalyzed by glutamine phosphoribosyl pyrophosphate amidotransferase, which replaces the phosphate chain with an amine group. This is the first committed step in the de novo purine synthesis. Glycine and ATP are then used and catalyzed by synthetase to produce 5-phosphoribosyl glycinamide, building on the amine group. 4-myltransferase uses N10-4-myl tetrahydrofolate to catalyze the product synthesis into 5-phosphoribosyl N4-myl glycinamide, adding an aldehyde group and creating tetrahydrofolate in the meantime. Despite inhibitors of the purine cycle being toxic to all dividing cells, this toxicity can be used against cancer as a form of treatment. Tetrahydrofolate is required in purine synthesis, thus folic acid analogues can be used to limit its quantity and slow DNA replication. Glutamine, ATP, and water are used alongside the synthetase enzyme to produce 5-phosphoribosyl N4-myl glycinamidine, which again by synthetase is aided with ATP use to create 5-phosphoribosyl 5-aminoimidazole. Carboxylase uses carbon dioxide to form 5-phosphoribosyl 5-aminoimidazole 4-carboxylate. This carboxyl group is then built on by synthetase, which uses aspartate and ATP to create 5-phosphoribosyl 4-N-succinylcarboxamine 5-amidoimidazole. Adenylose-succinate lyase uses water to synthesize 5-phosphoribosyl 4-carboxamide 5-aminoimidazole, producing fumarate as a second product. 4-myltransferase once again uses N10-4-myl tetrahydrofolate, producing tetrahydrofolate alongside 5-phosphoribosyl 4-carboxamide 5-formylamidoimidazole, which is then catalyzed by cyclohydrolase to remove a water molecule and produce inosine 5-monophosphate, also known as IMP. At this stage, IMP is synthesized into either adenine or guanine in the forms adenosine monophosphate and guanosine monophosphate, named AMP and GMP. These end products inhibit the synthesis themselves, so if there are more than one, then the other will be synthesized, and if there are adequate amounts of both, then the pathway is inhibited at the amidotransferase step, so as to not waste resources over producing them. For AMP, aspartic acid and guanosine triphosphate are used by adenylose-succinase synthetase to form adenylose-succinate, which is catalyzed by adenylose-succinase to form AMP, the end product. For GMP, water and nicotinamide adenine dinucleotide are used by IMP dehydrogenase to form xanthocene monophosphate, which is then catalyzed by GMP synthetase alongside ATP and glutamine to form GMP, the end product.